Hi, thank you for inviting me to this conference. I'm going to tell you about a story that we published this spring about inflammation activated by monocyte infection of SARS-CoV-2. This doesn't. Okay. A little delayed. Okay, so I'm going to tell you about what is arguably the most inflammatory immune response in the body, and it's called pyroptosis, for fiery inflammatory death, and it's triggered by invasive infection and danger signals. And it's not a transcriptional response. It has to be you have to express the molecules, which is regulated by transcription. But this pathway wouldn't be detected by the kinds of RNA sequencing that people are doing nowadays. So pyroptosis is mostly activated at sites of the body where we encounter infection, in particular in immune sentinel cells, like monocytes in the blood, macrophages in the tissue, dendritic cells, and they're activated by cytosolic sensors called inflammasomes. Unlike the toll-like receptors and other receptors on the outside of the cell, these sensors can distinguish pathogens from commensal infection. And the inflammasomes activate a family of caspases, the inflammatory caspases. And when they're activated, they have two functions. They process the IL-1 family cytokines into their active forms. And the IL-1 family cytokines, which are largely responsible for fever, have no secretory signals, so they are only secreted in the context of this inflammatory cell death. And the other event that the inflammatory caspases are responsible for is activating a family of pore-forming proteins in the cytosol called gasdermins for their tissue distribution in the skin and gastrointestinal tract and other mucosa, as well as in the myeloid sentinel cells. And those when they're cleaved by the caspases, they go to the cell membrane, form pores, which damages the membrane and causes cell death, and also allows the inflammatory cytokines to leave the cell. So we were very interested in whether the cytokine storm and severe COVID symptoms might be triggered in part, at least, by this inflammatory pathway. And sort of the molecular basis of this pathway is really only recently been described within the last 5 or so years. And there have been very, very few studies of pyroptosis and inflammasome activation by viruses. So I encourage all of you to think about whether you might want to study it in the context of viral infection, because most virologists have focused on interferons, because they have such an important role in antiviral immunity, but a lot of viruses counteract the interferon response. So our hypothesis was, since inflammaging and the comorbidities associated with severe COVID are associated with chronic inflammation, that perhaps pyroptosis might be one of the contributing factors for severe COVID. And we wanted to know, if so, how would it be induced and what cells might be responsible? So this is a cartoon that describes inflammasome activation and pyroptosis in myeloid cells. It starts with the sensor protein called an inflammasome in the cytosol, which recognize different variety of pathogen or danger-associated molecular patterns. When the sensor binds to one of these signals, it recruits an adapter called ASC and assembles into a very large complex called the inflammasome, which is about a micron in size. The inflammasome recruits the pro-inflammatory caspase-1, which gets activated in the inflammasome, and there it has these two functions I told you about, activating the pore-forming protein gasdermin D in myeloid cells and the pro-IL-1 family cytokines. So this work was described in a paper which was published this spring, and the people who did the work were Caroline Junquera and Angela Crespo in my lab, and Shaheen Ranjbar in my colleague Ann Goldfeld's lab, but it required a large community of clinicians and other researchers who are listed in the author list. So in order to do this study, we looked at whole blood, not frozen cells, cells that are immune cells that are activated or infected generally don't survive freeze-thawing. So it was critical that we looked at whole blood, and we wanted to know, was there an increase in pyroptosis or inflammatory death in the blood, in any of the immune cells in the blood? And to do this, we stained on the left with a small molecule dye that could be fixed, so we could work with COVID samples called zombie yellow that gets taken up by cells whose plasma membrane is damaged, and for annexin 5, which is a marker of classical apoptosis. And of all the immune cells in the body, we saw an excess of cell death, namely zombie uptake in the monocytes, if you look at the right graphs, but no increase in classical apoptosis or annexin 5 staining. And the top row are the healthy donor, and the bottom is a typical COVID-19 sample. Then we wanted to know whether there might be an increase in this inflammatory cell death in COVID patients, and we looked at markers that are more specific for pyroptosis than other markers, namely cleaved gasdermin D gets released during pyroptosis from myeloid cells. LDH is characteristic of any kind of necrotic cell death, and pyroptosis is one of the most common forms of necrotic death in the cell. IL-1 beta, the IL-1 receptor antagonist, which is induced by IL-1, as well as IL-18. And what you can see in the top row is that these markers were increased in the plasma of COVID patients compared to healthy donors. And if we looked at the bottom graphs and compared patients that were either healthy donors who were uninfected with people with mild, moderate, or severe COVID, you can see that these markers tended to be elevated with more severe disease. Then we wanted to know, could these monocytes actually have evidence for activated inflammasomes? And we stained blood cells for the adapter ASC for activated caspase-1, which is called a FLICA assay. And we found in the bottom row in the COVID patients that they had these large puncta of ASC specks, which are characteristic of inflammasomes, which co-localized with activated caspase-1. And what we looked at several different inflammasomes, and we found that two inflammasomes, NLRP3, which recognizes plasma membrane damage, as well as AIM2, which recognizes cytosolic DNA, were activated and formed ASC specs, but that an inflammasome that recognizes bacterial toxins was not. And then we wanted to know, were these cells that had inflammasome activation undergoing inflammatory cell death? And if you just look at the right-hand images, you'll see in healthy donors, there's no uptake of that zombie dye, and gasdermin D, which is normally in the cytosol, is diffuse in the cytosol, whereas in the COVID-19 samples, you see these characteristic balloons of the cell membrane, which are pathognomonic for pyroptosis. You see zombie dye uptake, and that gasdermin D has localized to the membrane. So the monocytes are undergoing pyroptosis. And then we wanted to know what activates the inflammasome activation. So we stained the monocytes from healthy donors or COVID patients. We co-stained them for ASC specs and for COVID nucleocapsid on top, or J2, which is an antibody which stains for double-stranded RNA, which is a sign of active viral replication. And basically, every cell that had an inflammasome was infected, and every infected cell had inflammasome activations. So basically, the monocytes that were undergoing pyroptosis were infected. They started to replicate the virus. Then we looked at autopsy specimens from people who died of trauma or COVID-19, and we stained for tissue macrophages with CD14. We stained for ASC specs in red, and for nucleocapsid in green. And basically, there was inflammasome activation in macrophages only, and even the infected epithelial and endothelial cells had no evidence of inflammasome activation. So tissue macrophages as well as blood monocytes had evidence for pyroptosis. And when we quantified it, we found that unlike in the blood, where every cell that had an inflammasome was infected, in the lung, the infected macrophages had inflammasome activation, but a quarter of all lung cells had evidence of inflammasomes activation, which is incredibly high. But there were no inflammasomes detected in epithelial and endothelial cells, and we don't know why. Monocytes don't express ACE2. They weren't thought to be infected. The question was, how did they get infected? Do they replicate the virus? And do they produce infectious virions? And what we found is that in the blood of COVID patients, only the non-classical monocytes that express CD16, the Fc receptor for antibody uptake, became infected. And usually in most patients, in most healthy people, that's about 10% of the monocytes, but that number is increased in COVID. And basically I have to hurry because I'm running out of time. And it turned out, so then we looked at healthy donors to see if we could infect their monocytes with SARS-CoV-2, and we found we could infect them, but only in the presence of a source of anti-COVID antibodies, either monoclonals or patient plasma. And if we depleted the plasma of antibodies, they did not become infected. We found that the infection was mediated by CD16 and was independent of ACE2. But one thing that was reassuring was that not all antibodies facilitated infection. So we didn't find infection at all with vaccinated donor plasma, even though their titers were twice as high on average as the patients with COVID, which facilitated infection. And if you're interested, I'll tell you what I think is going on there in the questions. And what's interesting is that even though the virus started to replicate, which we had shown by staining for double-stranded RNA, we verified that by amplifying subgenomic RNAs that are only produced by the virus during active replication. We found that there was no infectious virus in the culture supernatants. So pyroptosis is a very fast process. It occurs within minutes. The viral life cycle takes about 12 hours, and what we think is that the virus starts replicating, it activates pyroptosis, but it doesn't have a chance to finish the process because pyroptosis intervenes. And so that's my summary. We found that patients with severe COVID had more evidence of inflammatory cell death in their blood, that about 6% of monocytes became infected through antibody opsonized uptake via CD16. Not all antibodies mediate infection. And we also found a very high frequency of inflammasome activation in the lung of COVID patients. It's human tissue macrophages actually express both ACE2 and CD16, and work published back-to-back with ours from Richard Flavell suggests that both ACE2 and CD16 are involved in COVID infection of macrophages in a humanized mouse model. And what he showed in that paper, which we were co-authors of, was that you could inhibit NLRP3 or caspase-1 with available small molecules, and the lung pathology in the mice was greatly reduced. So inhibiting pyroptosis might prevent severe COVID disease. So the conclusion is that pyroptosis is really a double-edged sword. It aborts production of infectious viruses from monocytes and macrophages and helps stimulate protective immunity, but it can also trigger lethal inflammation. Thank you.
That was a great talk, Judy. I think what we might do is take one question, and then we'll take the rest of the questions during our discussion so that we can stay on time.
Sorry.
That's okay. It was a terrific, a lot of information. But so go ahead with the question.
Hi, it's Len Calabrese from Cleveland Clinic. I've really admired your work since you published it. So everybody makes antiviral antibodies who has COVID-19, yet this happens in a small population. Do you posit that this is due to some variation in the antibody, like glycosylation pattern or something? Or have you looked at FC genotypes to see if this is an influence?
Yeah, that's a great question. So, so in our paper, we provided evidence that was suggestive, I don't think it's definitive, that that antibodies that are afucosylated in their FC region facilitate infection. And it turns out that afucosylated antibodies have been linked in several papers to the development of severe COVID, but people didn't understand why. And CD16, the receptor that we implicated in this process, has about 10-fold higher affinity for CD16. We also did a study with a bioinformatician in London, which wasn't in our published paper, but was in the preprints, where we found that people who had EQTLs or SNPs that lead to higher expression of different inflammasome-related genes were more likely to develop severe COVID. So there may be some genetic links. Clearly, the people who get severe COVID have more chronic inflammation. Thank you.